All right, can we start?
Yes.
How about the others? Please open your cameras, please. Pagpasensya nyo na po, no? Medyo inuubo po ako. Every time po yan na nag-lecture ako, lagi po ako may sakit. Anyway, kita po yung screen. Can you see the screen?
Yes po, Doc.
Yes po, Doc.
All right. Okay, let's start. So for this afternoon's lecture, we'll be discussing porphyrins, RBC, and respiration. So again, I'm Dr. Don Jerome Santo Domingo, your lecturer for this afternoon. So just like in CBL, no, and in any other lecture, no, we need to first present the learning objectives because this will be your guide, no, in reviewing for the exams. So here are our learning objectives. Kung nababasahin isa-isa, no. Sige. First two is to describe the pathway of heme synthesis and identify the major organs or tissues that have the highest rates of heme biosynthesis. At any point during our discussion, you can ask questions, you can raise your hands, and then we'll try to answer it later on. Okay, so let's start. What is porphyrin? No? So porphyrin, wait lang. Makita ko yung mga mukha. Ayusin ko lang. So porphyrin, no, is a cyclic compound. No? It's a circular structure made up of 4 pyrrole rings. Kita niyo dyan, no? They are connected to methylene bridges. You can see there, complex with a metal ion. And in the case of heme, it's complex with iron. Okay? Okay, so your porphyrin ring, no, can actually have 4 additional bonds, or actually 6 additional bonds, 4 with nitrogen atoms, no, and 2 with oxygen, as well as the histidine residue in the globin chain. Later on, we'll be dealing more about those oxygen and histidine binding of your heme. So other porphyrins, no, as you see there in the slide, no, meron silang other metals that can be complex to it. One is your chlorophyll, no, the one that gives the plant its green pigment, complex with magnesium ion, and your cyanocobalamin, which is a vitamin complex with cobalt, no, in its center. So how do we number the porphyrin rings, no, the 4 pyrrole rings? So we can use Roman numerals 1 to 4, no, to number them, starting from the top, proceeding in a clockwise manner. Dito makikita nyo, pwede rin yung alphabet, no, A, B, C, D, no. So we can use Roman numerals 1 to 4 from the top, proceeding in a clockwise direction, no. And the methylene bridges, no, the one that connects your pyrrole rings, no, are designated with Greek letters, no. Again, designated as alpha to delta, proceeding in a clockwise manner then. So these are your methylene bridge, alpha, beta, gamma, and your delta. So the carbon atoms and nitrogen atoms in your porphyrin nucleus is numbered from 1 to 24, no? The peripheral carbons, no, are numbered 1 to 20, and the nitrogen atoms inside is given locants 21 to 24. Your porphyrin nucleus, no, can be substituted, or you can add side chains to your porphyrin ring at 2 positions, no? 8 of which is seen in the beta position, located in the pyrrolic rings. Excuse me. So that's 2, 3, 7, 8, 12, 13, 17, and 18. And 4 additional, no, you can actually add side chains, no, to the meso position, 5, 10, 15, 20, located in your methylene bridges. Okay? And please remember that a porphyrin molecule that is unsubstituted, walang kahit anong side chain, is known as your porphin. Okay? Porphin. Saka kasunod po, nasusundan. Okay. All right, let's proceed. So these are the common side chains, no, that you can attach to your porphyrin molecule. You can attach an acetate, no, or carboxymethyl or acetic acid, propionate or propionic acid, or carboxyethyl. You can have methyl side chain as well as vinyl side chain. On your right, you see the different porphyrins important in humans. So you have your uroporphyrinogen that has 4 acetate and 4 propionate side chain. You have your coproporphyrinogen, which has 4 methyl and 4 propionate side chain, and your protoporphyrin 9, which has 4 methyl, 2 propionate, and 2 vinyl side chain. Later on in the heme synthetic pathway, we'll be discussing how the side chains are formed. All right. So the nature and distribution of the side chains would determine the physical properties of your porphyrin, specifically its solubility or its polarity. So that the more carboxyl group present in your side chain, the more water soluble, the more polar that porphyrin is, no? And that you can classify your porphyrin into 4 types, no? We're in type 3 porphyrins, no? Ito lang yung concern tayo, no? Type 3 porphyrins is the most important porphyrins in human, and we can identify it by the presence of an asymmetric side chain in ring D. So di ba sabi natin kanina, the porphyrin, the tetrapyroles, the rings, no, can be labeled with Roman numerals 1 to 4, pwede ring A, B, C, D, no? And in ring D, no, the side chains is said to be asymmetric, no? Totoo ba yun? So this one is an example, your uroporphyrin 3, no, it's a type 3 porphyrin, acetate propionate, acetate propionate, acetate propionate, but in the ring D, it's asymmetric. Baliktad, no? Propionate, acetate. Can you follow? Susundan? Clear pa naman po. Sige. So don't be confused. Sometimes type 3 porphyrins are also known as your type 9 porphyrins. No? Okay. So let's now discuss heme. No? So heme is a flat planar structure containing porphyrin ring complex with iron on its center. No? And because of this iron, no, it allows heme to transfer and receive electrons. No? Transfer and receive oxygen. And most of the time, this heme, no, acts as a prosthetic group, no, in different proteins. No? So they are usually attached to proteins. And we call them heme proteins. No? Sorry. So heme proteins, depending on the 3-dimensional structure of the protein where your heme is attached, that will dictate the function of that heme protein. For example, your hemoglobin, no? So by now, for sure, you're familiar with hemoglobin or the hemoglobin molecule. So it's made up of 4 subunits of globin chains, right? So the main function of your hemoglobin is to transport oxygen in the circulation. Myoglobin is made up of 1 subunit of globin chain, no? When heme is attached to it, it can function as a storage of oxygen, no? Kasi mag-isa lang siya, no? It's not efficient in transporting oxygen. Only storage. Cytochrome C, which is a protein seen in your electron transport chain, no, for the transport of electrons. Your cytochrome P450, which is an enzyme produced in the liver for the hydroxylation of xenobiotics. No? So these xenobiotics are actually compounds, no, drugs, no, whether it's obtained exogenously or endogenously, like your steroids, no. When we hydroxylate them using cytochrome P450, we convert them into a more polar structure, no? So we can easily excrete them. Next is your catalase. No? Catalase is also a heme protein that is responsible for degradation of your hydrogen peroxide to water and oxygen, and your tryptophan pyrrolase for the oxidation of your tryptophan. Okay? Any question up until this point about porphyrin, heme proteins? Clear naman po so far? Sige. O, yung kakalimutan mo lahat yan, i-apply natin mamaya, no? So, huwag kayong mali dito. Alright, so let's now proceed with the next learning objective, heme synthesis. So, heme synthesis, most of the body's heme synthesis occurs in 2 organs, no? Your bone marrow, as well as your liver or hepatocytes, no? Where we produce heme at a rate of 0.3 grams per day, no? 80% of which happens in your bone marrow, no, for the synthesis of heme as part of your hemoglobin. No? This specifically occurs in the erythrocyte precursor cells. Again, I repeat, in the erythrocyte precursor cells. No? And 15% is in the liver, no, where it contributes for the production of cytochrome P450. Di ba ang cytochrome P450 is a heme protein containing heme? So 15% ng heme synthesis is in the liver. Okay, can you guys follow? Why did I mention erythrocyte precursor cell? Because heme synthesis is only one of the few pathways in our body that utilizes two cellular compartments. One is the mitochondria, and the other is your cytoplasm. Okay? So again, kasi di ba ang mature RBC ba? Does mature RBC contain mitochondria? No. So definitely you cannot synthesize heme in a mature RBC. So usually yan sa mga precursor RBC, pro-erythroblast, pro-normoblast. No? So yung mga may mitochondria lang. Alright? Okay. So this is just an overview of your heme synthetic pathway. And as I have mentioned, it is one of the few pathways that utilizes both the mitochondria and the cytoplasm. In fact, the initial step of your heme synthesis occurs in the mitochondria, the intervening steps occurs in the cytoplasm, and the terminal steps go back again to the mitochondria. The other pathways that utilizes your mitochondria and cytosol include your urea cycle, as well as your gluconeogenesis. Okay, so the one that we'll discuss is heme synthesis. So when I was reviewing for the boards, no, I only remember the mnemonic hug, no. So it's like the mitochondria and the cytoplasm hugging each other, no, for them to carry out these pathways. All right. Please remember that at elevated concentration, intermediates of the heme biosynthetic pathway is toxic. No? Ano ba ang goal? What's our goal in heme synthetic pathway? For us to produce heme, so that any intermediates that we'll be producing are all toxic. Okay, please keep that in mind. So applying what we learned about the side chains a while ago, okay, let's try to analyze. Amongis, Archie, are you there? Amongis, Archie. Bawal po ang Zoom party, no? Kung Zoom party po, wala po sa akin. Okay lang, wag na po ng time mag-attend. Don't attend the class.
Hello
Instead,
po.
just wait for the... Oh, hello. Yes, Archie, are you there?
Yes po, Doc.
Okay, so applying what we learned about the side chains a while ago, between your protoporphyrinogen and uroporphyrinogen, which is more water soluble?
Is it the protopol, Doc?
Yeah. Do you agree?
No, sure po.
Linnea? Sige, okay lang. Do you agree, Linnea?
No po, Doc.
Okay, so what's your answer?
No po, Doc.
So if you don't agree, what's your answer?
Yung uroporphyrinogen po.
Okay, and why is that? Bakit mas water soluble ang uroporphyrinogen? Yan ang sabi ng katabi mo, Linnea. Sige, si Mena.
Doc, dahil nasa cytosol and may mas madami yung hydrogen niya.
Okay, that is because of the carboxyl group, no? As you can see here.
Yes po.
Madami ba siyang carboxyl group
Carboxyl.
versus your protoporphyrinogen? Mas madami ba? Ang sabi natin kanina, the higher the number of carboxyl group, the more water soluble that porphyrin is. Tama ba? Meron bang clinical relevance yun? Bakit mo ba sinasabi yan sa amin, Doc? Parang masyadong mambayotan yan. Meron po, no? So that when we receive patients that we suspect, no, porphyrias, no? Later we'll be discussing porphyrias, no, the conditions related to defective heme synthetic pathway. If you want to isolate your protoporphyrinogen, where would you isolate it? In the urine or in the stool?
Urine po.
Sino ang mas water soluble? Sino ang mas fat soluble or more non-polar? So definitely it's your protoporphyrinogen. So you'll see more of your protoporphyrinogen in the stool, no? In comparison, your uroporphyrinogen, kaya nga yan ang naging pangalan niya uroporphyrinogen. It's initially isolated in the urine, no? So because of its polarity, no, your uroporphyrinogen is mainly seen in the urine. Okay? Nasundan po? Alright. Alright, Brink, you're raising your hand.
Yes, Doc. So why I didn't get the part where all intermediates are toxic?
Yes, later on we'll be discussing why the intermediates are said to be toxic. One of the reasons here is some of the intermediates can cause damage to the neurons, and some of the intermediates, when exposed to light, when they are exposed to light, they can create reactive oxygen species that can damage the skin. Later on, when we discuss the porphyrias, we'll be discussing why these intermediates are toxic.
Thank you, Doc.
All right. So let's start with the first step of your heme synthetic pathway, formation of delta aminolevulinic acid or D-ALA. So this step happens in the mitochondria. Di ba sabi natin kanina? This is through the condensation of succinyl-CoA, an intermediate in your Krebs cycle, and glycine, no? Through the enzyme ALA synthase to form your D-ALA or delta aminolevulinic acid. ALA synthase is the rate-limiting enzyme of heme synthetic pathway. O mula ngayon ha, dapat alam lahat ng rate-limiting enzymes ng bawat metabolic pathway. Mortal sin po yan na hindi natin yan alam, no? Okay? It's a must-know. So ALA synthase is a decarboxylating enzyme. So this leads to the release of coenzyme A and carbon dioxide. This enzyme also requires a coenzyme known as your pyridoxal phosphate, PLP. This pyridoxal phosphate is actually derived from a vitamin, which is vitamin B6, pyridoxine. All right. So that patients with vitamin B6 deficiency, can they develop anemia?
Yes po.
Yes, Doc.
Yes, no? So, kasi as you see here, the rate-limiting enzyme of heme synthesis, no, eventually will be incorporated to hemoglobin, requires pyridoxal phosphate. So definitely, they can develop anemia. Okay? What else? So your ALAS, has two isoforms, no? One is your ALAS1, which is also known as your housekeeping type. No? This is encoded in chromosome 3. Okay? While the other enzyme, ALAS2, is restricted to the erythrocyte precursors. No? And this is encoded in the X chromosome. Please remember that synthesis of ALAS, no, or expression of ALAS requires iron, because the mRNA of ALAS contains iron response element, no. So you need iron for its synthesis. So iron is a positive regulator of the enzyme. It will stimulate ALAS. Okay, does that make sense? Yes, kasi heme, if you remember, does heme contain iron? Yes, it contains iron on its center. No? So iron definitely stimulates its synthesis. While presence of heme as well as glucose, heme is the end product of this pathway. Diba? Heme synthesis, end product heme. Heme being the end product would cause negative feedback inhibition to the rate-limiting enzyme ALAS. Okay? Glucose also inhibits ALAS. Later on, we'll be discussing what's the mechanism behind that. Okay? So next, this ALA no, the D-alad that you form will go outside of the mitochondria to the cytoplasm, and two molecules of which is needed, no, to form your porphobilinogen, the first precursor pyrrole. Sabi natin kanina, di ba, ang porphyrin, ilan siya? It's made up of 4 pyrrole rings. So gagawin natin yung first precursor pyrrole, the first pyrrole ring. And this is catalyzed by the enzyme ALA dehydratase, sometimes called as ALA dehydrase. No? This is a dehydrating enzyme. As you can see, there is a release of water, no, in the process. Human alad, and another thing to remember, human alad is a homooctamer. It's made up of 8 subunits that is capable of binding zinc. No? It can bind 8 zinc atoms. 4 of those octamers requires sulfhydryl groups for its enzymatic activity. And presence of heavy metals like lead, no, can actually chelate, no, or remove, no, the zinc atoms from this enzyme, rendering this enzyme inactive. Can you follow? Sundan po? Okay. So now, porphobilinogen. How many molecules of porphobilinogen for you to make a porphyrin? Kailangan mo ng 4. You need 4, right? So 4 molecules of porphobilinogen would condense, no, to form your linear tetrapyrrole, otherwise known as your hydroxymethylbilane, catalyzed by the enzyme porphobilinogen deaminase, also known as hydroxymethylbilane synthase, because you're synthesizing hydroxymethylbilane. Please take note of the side chain that you produce, no, from your hydroxymethylbilane. It's made up of acetate and propionate. Okay? Tandaan, para hindi malito dun sa mga side chains. This hydroxymethylbilane has 2 fates. No? It can either undergo spontaneous cyclization, meaning non-enzymatic, to form your type 1 uroporphyrinogen, or enzymatically using uroporphyrinogen 3 synthase to form your type 3 uroporphyrinogen. Again, what's the difference? How will you identify a type 3 porphyrin, Vidal? Please acknowledge kung narinig na tinawag.
Yes, Doc.
So how will you distinguish a type 3 porphyrin again?
Doc, may I try
Doc,
po?
may asymmetric side chain po.
Okay, so correct. Thank you, Arsinas. I appreciate now your. So correct, no. It's it's the presence of an asymmetric side chain on ring. Kailangan specific on ring D.
Di po.
On ring D or the 4th ring, no? Totoo ba yon? As we can see here, acetate propionate, acetate propionate, acetate propionate. Sa ring D, baliktad na. Propionate, acetate. Nasusundan? While in the spontaneous cyclization, it contains a symmetric side chain. Okay? So what happens next? So we have your uroporphyrinogen 3, no? Which would undergo decarboxylation, converting the acetate side chains to methyl side chains. Okay? So this would considerably decrease the number of carboxyl group, no? Decreasing the polarity of your porphyrin, no? So di ba ito, 8 yung carboxyl group niya. Pagdating sa coproporphyrinogen, you only have 4 carboxyl group. And methyl, no, is a side chain known for its non-polarity. No? So it's more fat or lipid soluble. Can you guys follow? So pag pinag-compare natin, which is more water-soluble between uroporphyrinogen and coproporphyrinogen, definitely it's the uroporphyrinogen, right? So as I've mentioned, no, the enzyme uroporphyrinogen decarboxylase is a decarboxylating enzyme. So you have release of 4 molecules of carbon dioxide. And please remember, no, must know, uroporphyrinogen decarboxylase is the most commonly mutated enzyme, no, in the heme synthetic pathway, no, leading to the development of porphyria cutánea tarda that we will discuss din naman later on. As you can see here, no, the carboxyl group from your uroporphyrinogen is converted to methyl side chain. All right. So this is the last step, no? This is the last step in the cytoplasm because your coproporphyrinogen, no, would go back inside the mitochondria through your ADCB6 transporter to undergo 2 rounds of oxidation. Initially, via your coproporphyrinogen oxidase to form your protoporphyrinogen 9. Sabi natin, 3 and 9 are the same, no? So your protoporphyrinogen, protoporphyrinogen 9, you will have conversion of some of the propionate side chains to your vinyl side chain, again decreasing the polarity of your porphyrin. Okay? So, next round of oxidation from protoporphyrinogen 9 to your protoporphyrin, utilizing protoporphyrinogen oxidase, using 3 molecules of water with the release of 3 molecules of hydrogen peroxide. Okay. So what else would I like you to remember about the different porphyrins? Porphyrinogens, tandaan, magkakalimutan, must know. Porphyrinogens can undergo spontaneous oxidation to their porphyrin counterparts. What do I mean by that? Kahit wala yung protoporphyrinogen oxidase, protoporphyrinogen can spontaneously oxidize to its protoporphyrin counterpart. Uroporphyrinogen, even without an enzyme, can auto-oxidize to your uroporphyrin, and coproporphyrinogen can undergo spontaneous oxidation to coproporphyrin. Later, malalaman nyo what's the relevance, no, of this auto-oxidation. Another question, what did you notice, no, with the oxidation of protoporphyrinogen 9 to its protoporphyrin? What happens to its structure? Nixon, Lee?
Yes po, Doc. Nawalan po ng hydrogen po.
Nawalan ng hydrogen, saan?
Based on the structure po. In the ring.
Okay, so nabawasan ng hydrogen atoms, and in turn, anong nangyari sa kanya? Take a look at the methylene bridges.
Karoon po ng double bonds pa.
Correct. So as you see here, the methylene bridges, no, are converted to methine bridges, which contains double bond. May significance ba yon? Yes, no? Because presence of double bond allows the porphyrin to absorb light and eventually display color. So ito po yung biochemical explanation why our RBC, why the hemoglobin is red in color. Because the protoporphyrin, no, present in it can absorb light and display color. Okay? Nasundan po. Later on, matututunan din natin that when these porphyrins are exposed to light or to UV rays, no, the reaction can release reactive oxygen species. Tatandaan niyo po yan, ha? No, porphyrins, when exposed to UV light, no, can generate reactive oxygen species. And these are damaging compounds, no? Titingnan natin later what's the significance. Okay? So let's try to differentiate porphyrinogens with porphyrins. Porphyrinogens, no, are the reduced form, while porphyrins are the oxidized form. Porphyrinogen contains methylene bridges, while porphyrin contains methine bridges, no, double bonds. Porphyrinogens are colorless, while your porphyrins are colored. Since they are reduced, no, your porphyrinogens are sensitive to oxidation, so there can be auto-oxidation or spontaneous oxidation of porphyrinogens to porphyrins, while porphyrins, no, is capable of reduction, and syempre, given naman yung last sentence. Right? So the last step of your heme synthetic pathway still occurs in the mitochondria, no? And that is through the incorporation of iron to your protoporphyrin 9 catalyzed by your ferrochelatase, no? Forming now your heme. This heme can then be transported out of the mitochondria to be used by different proteins to form heme proteins. Sabi natin kanina, depending on the proteins where it will be attached, no, that will determine the function of that heme protein. So itong nasa taas, this is a hemoglobin, no, where your heme can act as a prosthetic group. Ito naman, this one is a myoglobin. What else do we need to remember? Iron, as you can see here, is a positive regulator of ALAS. When we say positive regulator, it stimulates the ALAS. Bakit? Why? As I mentioned a while ago, the mRNA of ALAS contains iron response element, no? Okay, what else? Heme, the end product of the pathway, no, present in cytochrome P450, is a negative regulator or inhibitor of your ALA synthase. Okay, what else? Ferrochelatase, just like your ALA dehydratase, is a zinc-requiring enzyme that is inhibited by presence of lead. Okay, so when you're asked in the exam, what are the enzymes in the heme synthetic pathway that is inhibited by lead? Sagot, ALA dehydratase and your ferrochelatase. Okay, ano pa? What do I want to emphasize? Ferrochelatase, aside from incorporating iron to heme, can also incorporate other metal like zinc, no, to your protoporphyrin, forming your zinc protoporphyrin. Okay? And protoporphyrin that is unbounded to any metals, no, if it's existing in a free form, is known as your metal-free protoporphyrin. Because we will be encountering these terms, no, when we discuss the different porphyrias, no? So I'm emphasizing it. All right. So just a brief summary of everything that we have discussed. As I've mentioned, your heme synthesis occurs in two cellular compartments, mitochondria and cytoplasm. The initial pathway occurs in the mitochondria with the condensation of glycine and succinyl-CoA through your ALA synthase, the rate-limiting enzyme, requiring pyridoxal phosphate derived from vitamin B6, forming your delta-aminolevulinic acid, two molecules of which would go outside of the cytoplasm to be catalyzed by ALA dehydratase or ALA dehydrogenase, no, to form your porphobilinogen, the first precursor pyrrole. Four molecules of porphobilinogen would then condense by PBG deaminase or hydroxymethylbilane synthase to form your linear tetrapyrrole, your hydroxymethylbilane. Hydroxymethylbilane can undergo spontaneous cyclization, no, forming your type 1 uroporphyrinogens, or through the enzyme uroporphyrinogen 3 synthase to form your uroporphyrinogen 3. Uroporphyrinogen 3 would undergo decarboxylation by urod or uroporphyrinogen decarboxylase to form your coproporphyrinogen, converting the acetate side chains to methyl side chains. Coproporphyrinogen 3 will go back in the mitochondria to undergo series of oxidation, initially to your protoporphyrinogen, converting some of the propionate side chains to vinyl side chains. Protoporphyrinogen 3 would undergo another oxidation by protoporphyrinogen 3 oxidase, forming now your protoporphyrin 9. Protoporphyrin 9, when you add iron with the help of ferrochelatase, you will form now your heme that can be incorporated in proteins to form your heme proteins. In this example, it's your hemoglobin. Is it clear? Naintindihan po ba?
Can I ask a question, Doc?
Sure, go ahead.
I don't know how to pronounce it, a photoporphyrinogen. So I know for inactive enzymes, they end with gen, but what does the gen mean in this name?
Well, it's like a precursor. It's the porphyrinogen. No, it's the one that contains methylene bridge, no?
Okay.
The single bond. That when it's converted into its porphyrin counterpart, no, it will acquire double bonds, no, as I mentioned a while ago, that can absorb light, no, and generate reactive oxygen species.
Okay, sure. And what about what does the in the enzymes, what does the ALA stand for?
That's your ALA, or your aminolevulinic acid.
Okay.
Aminolevulinic acid synthase, because you're synthesizing delta aminolevulinic acid. All right. Questions?
Thank you, Bodo.
Clear so far? Kapit lang kayo, papunta pa lang tayo sa exciting part. So, wait lang.
Doc, question pala po.
Sige, go ahead.
I'm not sure if I missed it kanina, pero saan po galing yung globin chains pala?
Well, globin chains, later on we'll be discussing it, no? How we synthesize the globin chains, no? It's
Okay,
actually synthesized from a different gene cluster. So ito lang, we're discussing the heme component, no, of your heme proteins. Okay. Okay? Okay naman?
Excuse po.
Sige,
Tanong po
daan
doon sa
ka
ano?
muna.
Ano po ulit yung nakalagay na ANN doon sa Europorphyrinogen 3? Medyo nawala po ako sa part ko na yun.
Alin, sorry. A, this one. Diba, if you remember, what are the side chains that you see in your Europorphyrinogen? Diba, pinakita ko sa inyo yung structure nila kanina, yung mga examples. So, what are the side chains you see? The acetate and propionate. No? So, if you're going to your Europorphyrinogen originally contains acetate side chains, no? But by catalysis of your Europorphyrinogen decarboxylase, these acetate side chains, all four, no, are converted to your methyl side chain.
Okay, so, kuha po.
O, diba? 4 acetate and 4 propionate side chain. So dito, coproporphyrinogen 3 contains 4 methyl and 4 propionate side chain. Another oxidation would convert some of the propionate. How many propionate do you have? 4, diba? So some of the side chains will be converted into vinyl side chain and some will remain propionate. So that your protoporphyrinogen contains 2 vinyl, 2 methyl, 4 methyl, 2 vinyl, 2 propionate, and 4 methyl side chain. Okay?
Thank you, Doc.
Okay. So let's now proceed with the next objectives. So ito guys pag nag-aaral kayo in biochemistry, when you're discussing the different metabolic pathways, it's also paramount to know the different diseases related to it, no? Hindi lang naman tayo forever medical students, no? We'll be doctors. So for sure, we need to know what are the diseases related to this metabolic pathways. Okay? So yun yung sasagutin natin in our next learning objective. Describe how defective heme synthesis leads to porphyria. Okay, so porphyrias. Are you familiar with the person who is in the slide? Do you know this guy?
Abraham Lincoln.
Ha? Sino siya? Wala namang score to. Okay, sino kaya yan? May movie yan eh.
So sabi po kanina,
US President.
Abraham Lincoln daw po. May
Abraham Lincoln?
Di ba? Are you familiar with that movie, Abraham Lincoln Vampire Slayer? Napanood niyo na yung movie yun?
Yes.
Anyway, sabi nila, porphyrias daw, no, the medical porphyrias daw are the medical explanation for someone that is labeled as vampire. Why? Later on, we'll be discussing. First, ano ba yung mga characteristic ng vampire? Are they afraid of light? Takot ba sila sa araw? Yes, no.
Yes.
Some patients with porphyrias will learn later on, will develop cutaneous manifestations when they are exposed to UV rays, no, from the sun, no, sun exposure. What else? They are pale, right? Maputla. This is because now they have defective heme synthesis, no. What else? They have bizarre behavior. Tama ba? Behavior, bizarre. No, bakit? No, because some of the intermediates or some heme synthetic pathway defects, no, would give you neuropsychiatric manifestations. No, they can have depression, psychosis. No, so sagutin na yan natin later. Okay. First, what are porphyrias? They are rare metabolic disorders caused by altered activities of enzyme in the heme biosynthetic pathway. Porphyrias are generally classified into two groups, the hepatic, no, and the erythropoietic, depending on the organ primarily affected. Please remember, must know, wag kakalimutan hanggang maging doktor bago mag-board exam. Manifestations of porphyrias can either be two, no? One is neurovisceral. When we say neurovisceral, no, manifestations include abdominal pain, motor and sensory peripheral neuropathy, neuropsychiatric changes. Or it can be cutaneous manifestations, chronic and blistering manifestations, or acute and mostly non-blistering. So what's the reason? Bakit ganito? Why is this the manifestation of porphyrias? First, neurovisceral manifestations is said to be due to elevation on the earlier intermediates in heme biosynthetic pathway. That earlier intermediates, ALA, PBG. Ito ba yung mga unang na-synthesize natin? ALA from ALA synthase, porphobilinogen from ALA dehydratase. Because they said, based from literature, no, ALA, elevation of ALA and PBG is damaging to the neurons. So pag na-damage ang neurons, no, their manifestation is no, neural din, abdominal pain, no, pwedeng neuropathies, no, or in the central nervous system, you can have depression, psychosis, no. So yan. While cutaneous manifestation is said to be due to elevation of the later intermediates of the heme biosynthetic pathway. This patient, no, is a patient with porphyria cutaneous tarda. So you can see here, yung skin niya, no, has cutaneous manifestation. This is due to elevation of porphyrinogens and porphyrins. Basically, when you already have the cyclic structure, di ba? When you already have the cyclic structure. So, how can porphyrins, no, again, damage the skin? As I mentioned a while ago, it contains double bonds, right? That can absorb light. And when it absorbs light, it can generate reactive oxygen species. That when these porphyrins, no, traverses the superficial blood vessel, no, and they're exposed to the sun, no, it can damage your skin. Nasusundan? Okay. How about porphyrinogens? Why would porphyrinogen cause skin manifestation? Eh di ba nga yung porphyrins lang yung nagkakaroon ng light absorption, the generation of reactive oxygen species? Ulit-ulit lang. Tinitingnan ko lang kung naalala niyo. What's with porphyrinogens? Sabi nga natin, porphyrinogens can undergo spontaneous oxidation to their porphyrin counterparts. Tama ba? O, pag nag-spontaneously oxidize sila, magiging porphyrins na ba sila? Pag naging porphyrin sila, meron na silang double bond that can absorb light, generating reactive oxygen species. Clear? Uy, react naman kayo dyan.
Yes
Simple
po.
daw.
Okay, sige. Sorry, medyo masakit na yung lalamunan ko. No? Ang hirap palang mag-lecture, no? Dire-diretso, tapos may ubo pa ako. Sige, let's proceed. Now that you know the different effects of the different intermediates, no? Let's now proceed to the different types of porphyria. No? Hindi pa pala. So this is the answer back to your question a while ago, no? Intermediates of the heme synthetic pathway are said to be toxic, no? Okay, so let's just again review, no? Porphyrias are due to mutation in various genes, no, for heme synthetic pathway. Again, when your ALA and PBG accumulates, what's the manifestation? Neuropsychiatric, neurovisceral. When your porphyrinogens accumulate, they can undergo spontaneous oxidation to porphyrins, leading to photosensitivity. Right? So let's now go to the different types of porphyrias. Okay. First is your X-linked protoporphyria. X-linked protoporphyria no, is caused by a gain of function mutation. Pag sinabi natin gain of function mutation, you upregulate the enzyme. No? Okay? You upregulate the enzyme ALAS2. So this is an X-linked dominant, primarily affecting the RBC, the erythrocytes. Since it's X-linked dominant, no, males are severely affected. As I mentioned, this is due to upregulation of your ALA synthase. And because of this, the intermediates no, of your heme synthetic pathway would also elevate. Nasusundan, syempre active yung ALAs. Yung intermediates, syempre, they are also upregulated. So patients would manifest as photosensitivity due to increase in your porphyrins. No? They will have red cell and hemoglobin decrease no, or anemia because they said that X-linked protoporphyria patients have relative iron deficiency. No? So they have iron deficiency. So even if you have an overly active ALAS, if your iron is deficient, you cannot form heme. Okay? So this is manifested or laboratory test would reveal high total protoporphyrins, high metal-free protoporphyrin. What is metal-free protoporphyrin? Those are the protoporphyrins without iron or zinc attached to it. Why would they elevate? No, because of overproduction of the intermediates. Like for instance, your protoporphyrin. No, not every protoporphyrins are complex with iron. Not every protoporphyrin is complex with zinc. So the levels of metal-free protoporphyrin would also elevate. No, same is true for your zinc protoporphyrin. They will elevate. No, because not only that protoporphyrin will be complex to iron, a lot also will be complex to zinc. So 15 to 50% will be zinc protoporphyrin. Okay, next. Next is ALA dehydratase porphyria, otherwise known as your DOS porphyria or plumboporphyria. This is the rarest of all no, porphyrias. There are actually only 4 documented cases worldwide. No? So it's very unlikely for us to see this in our lifetime. So this is due to a severe reduction in the enzyme ALA dehydratase here. This is an autosomal recessive condition, primarily affecting the liver. But for unknown reason, this patient would develop also increase in coproporphyrin here, no? But when you're asked in the exam, what's the primary manifestation of someone with ALA dehydratase porphyria? Answer is neurovisceral manifestation. Bakit? When you inhibit the ALA dehydratase, what would elevate? ALA. Again, ALA is responsible for the neurovisceral manifestation. So there are conditions that can actually mimic ALA dehydratase porphyria. One is lead poisoning. No, we discussed that a while ago. Lead can actually inhibit ALA dehydratase, no, causing now or mimicking now the symptoms of ALA dehydratase porphyria. Patients with hereditary tyrosinemia type 1. Narinig nyo na yun? Hereditary tyrosinemia type 1. Saan na kayo sa protein metabolism, di ba? So, hereditary tyrosinemia type 1 is actually an inborn error in tyrosine metabolism. Naalala nyo, tyrosine can be converted to fumarate and acetoacetate. Ba pwede din yan? So, in hereditary tyrosinemia type 1, what's the defective enzyme? Fumarate, acetoacetate, hydrolase. When this enzyme is defective, fumaryl acetoacetate cannot be converted to fumarate and acetoacetate. Instead, it is shunted to a different pathway, leading to the formation of succinyl acetone. Take note, Succinyl acetone is a potent inhibitor of ALA dehydratase. So if you have hereditary tyrosinemia, what happens to your succinyl acetone? It will elevate. Will you mimic the symptom of ALA dehydratase porphyria? Yes. Okay. You have almost the same manifestation. Okay. Next is acute intermittent porphyria. In acute intermittent porphyria, the problem is your porphovirinogen deaminase here. So this is an autosomal dominant disorder, primarily affecting the liver. Okay. So what would happen here is that you will have elevation of your ALA and PBG. ALA and PBG. And sabi nga natin, pag ALA and PBG ang mag-elevate, what's the manifestation? Neurovisceral or cutaneous? Neurovisceral. Okay. So you have abdominal pain, neuropsychiatric symptoms. Excuse me. Next is your congenital erythropoietic porphyria. This is an inherited deficiency in your uroporphyrinogen 3 synthase. This is an autosomal recessive disorder primarily affecting the erythrocyte. Manifestations include photosensitivity and hemolytic anemia. Question is, why would patients with congenital erythropoietic porphyria develop photosensitivity? Ensho, Danica? Ensho?
Ano po yan?
Why would patients with congenital erythropoietic porphyria develop photosensitivity? Danica? Okay, wait lang, Margo, ha? Danica?
Doc, nag-disconnect po.
Nasasakyan pa ata si Danica. Ano to? Classic to, no? Kanina pa sa morning, no? Lahat ng tinatawag ko, lahat may internet problem. Sige. Sige, Timothy.
Hello po, Doc.
Sige,
Doc,
Timothy,
I
go
think
ahead.
the reason po, I'll just guess po, the reason why usually people with this condition have photosensitivity is due to the fact that usually kapag when one is exposed to UV, you're usually and when you have blood deformities, blood releases reactive species po. So and when you have that, it's usually have problems with like photosensitivity and all po, Doc.
Sige. So sabi natin a while ago, no, the photosensitivity is due to increase in porphyrinogens, no, the cyclic structure. But if you inhibit, no, your uroporphyrinogen 3 synthase, what's the intermediate before it? Hydroxymethylbilin. Tama ba? That's the intermediate before it, right? Is it a cyclic structure?
Sorry po, Doc.
We mentioned that in CEP, congenital erythropoietic porphyria, the problem is in the uroporphyrinogen 3 synthase. So definitely, the intermediate right before this enzyme cannot pass through. No? So we mentioned a while ago that photosensitivity is only observed if you have high levels of the cyclic structure, porphyrinogens and porphyrins. Is hydroxymethylbilin a cyclic structure?
Yes po, Doc.
Cyclic ba siya?
Linear.
Di po.
It's a linear structure, oh. Sige, Margo, I think you're raising your hand a while ago.
Oh, no po. It was just to answer your question po.
Okay. So, sige, anyone from the class, why would patients with CEP develop photosensitivity?
Oh, Doc, I thought sinagot na ni Timothy. Pero, Doc, the way I understand it, is it because there's an excessive non-physiological amount of Porphyrin,
Cor-
porphyrins.
Correct. Okay, that's correct, no? So, nakita niyo dito, no? The intermediate right before that enzyme is hydroxymethylbilane, right? Ba? And I mentioned a while ago that there are two fates, no, for hydroxymethylbilane. One is the non-enzymatic cyclization. Tama ba? Non-enzymatic cyclization leading to the formation of the type 1 uroporphyrins. Type 1 uroporphyrinogen and type 1 coproporphyrinogen. So even though you have a block, no, in this enzyme, no, you cannot produce your uroporphyrinogen type 3. There would be elevation in the non-physiologic porphyrins, no? Your uroporphyrinogen 1 and coproporphyrinogen 1 that is converted, no, or they will auto-oxidize to form their porphyrin counterparts. And when they become porphyrins, no, and they generate the active oxygen species, pwede na, no? This will lead to photosensitivity. And the fact that they are non-physiologic, no, this non-physiologic porphyrins can actually cause hemolytic anemia. Okay? Alright, naintindihan po? Okay. Next is your Porphyria cutánea tarda. Porphyria cutánea tarda is the most common type of porphyria, no? So the problem here is you have a defect or a deficiency in your uroporphyrinogen decarboxylase or UROD enzyme. No? So this condition would only manifest if the enzymatic activity is said to be less than 20%. No? So your uroporphyrinogen decarboxylase is very efficient, no, in converting your uroporphyrinogen to coproporphyrinogen. That if you have less than 20% activity, that's the only time that you will manifest the condition. Okay? So this leads to an increase in your uroporphyrins. No? Uroporphyrins would later on result into your blistering and skin fragility. No? So kaya siya tinawag na Porphyria cutánea tarda. No? So the manifestation is primarily in the skin. Okay. Next is your variegate porphyria. In variegate porphyria, the problem here is in your protoporphyrinogen oxidase. This is inherited in an autosomal dominant manner, primarily affecting the liver. So in this case, based from literature, they have elevation in urinary ALA, PBG, as well as your coproporphyrin 3 and protoporphyrin 9. So, yung manifestation niya is mixed, no? Tandaan nyo, isa lang to sa mga few porphyrias that manifest both neuropsychiatric, no, and cutaneous manifestation. Wait lang. For that problems. Anyway, so as I mentioned, it will manifest both neurovisceral and cutaneous manifestation. So sabi dito, based from here, you will have elevation in your protoporphyrin. The intermediate before your protoporphyrin oxidase is protoporphyrinogen 9. It cannot be converted enzymatically to protoporphyrin 9. Why would you have elevation in your protoporphyrin? Alexandra? Sorry, ulit-ulit na tayo, no? Talagang ine-emphasize natin. Why would you have elevation of protoporphyrin 9 if you have a block in this enzyme?
Doc, ano po ulit yung tanong po?
Sige. So in variegate porphyria, the problem is in your protoporphyrinogen oxidase. So yung enzyme na yon converts protoporphyrinogen to protoporphyrin. But sabi dito, you will have elevation of protoporphyrin 9. Why would you have elevation of protoporphyrin 9 when you cannot convert protoporphyrinogen to protoporphyrin?
Doc, sorry, hindi ko po alam yung sagot.
Sige. Sinong close friend mo dito, Alessandra? Call a friend.
Si Carl po. Reyes.
Sige. Carl? Okay. Carl?
Dok, hindi ko po siya friend.
Pinapil na.
Doc, pwede i-try ko na lang po.
Sige.
Para
Miss
sa
Abu.
is it because po, spontaneous yung pagkagawa nung so kaya niya
Yes.
pong
So,
gumawa. So kaya niya pang
Yes.
mag-form.
Okay, so that is because of the spontaneous oxidation of the porphyrinogens to their porphyrin counterparts, even without the use of enzyme. Diba? We mentioned that a while ago. So you would also have elevation in your coproporphyrin as well as your urinary ALA and PBG. Sabi nila, the reason for your urinary ALA and PBG elevation is because protoporphyrinogen, protoporphyrinogen is able to activate your ALA synthase. No? Your ALA synthase, again, would generate the intermediates ALA and porphobilinogen. But at the same time, your protoporphyrinogen inhibits another enzyme, your porphobilinogen deaminase. So ang end effect niya will be elevation of ALA and PBG, no, and at the same time, increasing your porphyrins. So that the manifestation is both neurovisceral and cutaneous manifestation. Okay. Next, hereditary coproporphyria. This is just like your variegate porphyria, no? Wherein you have elevation in ALA and PBG as well as your coproporphyrin. Okay. So in this case naman, the problem is in your CPOX, no, or your coproporphyrinogen oxidase. No? Kanina, the variegate porphyria is your protoporphyrinogen oxidase. But this is also a type of porphyria with mixed manifestation, neurovisceral as well as cutaneous manifestation. Okay. Lastly is your erythropoietic protoporphyria. This is a cutaneous porphyria characterized by painful, non-blistering photosensitivity as well as anemia. This is an acquired or inherited loss or decrease in function in your ferrochelatase. This is autosomal recessive, primarily affecting the erythrocyte, which will lead to an elevation in your protoporphyrin, in your metal-free protoporphyrin, as well as in your zinc protoporphyrin levels. So again, why would you have elevation in your metal-free protoporphyrin? Bakit magkakaroon ng elevation in metal-free protoporphyrin? Because you're lacking the enzyme, no, that adds iron or zinc, no, to your protoporphyrin. So protoporphyrin would exist in a metal-free form. So you'll have an increase in metal-free protoporphyrin. Would you still have zinc protoporphyrin? Yes, pero it's decreased, no, less than 15%. Why? Di ba you remember in your enzyme, no? Do you absolutely need an enzyme for you to make an end product? Hindi naman, di ba? So you can still make zinc protoporphyrin, no? But it's in very low amounts, less than 15%. Okay. So next. Okay. Any questions up until this point?
Doc, can I ask a question, Paul?
Sure, go ahead.
Is there any positive thing related to the spontaneous oxidation, since it's related to a lot of negative things, seems like it?
Not that I know of. The only thing I can think of is that if you have spontaneous oxidation of protoporphyrinogen to protoporphyrin 9, then you can have the this one. If you have spontaneous oxidation of protoporphyrinogen to protoporphyrin 9, then you can already have a substrate, no, where your ferrochelatase can add iron for you to form heme. Okay, but other than that, no, I think it's mostly related to generation of reactive oxygen species.
Thank you, Paul.
Okay. Sige. So, any questions? Okay pa ba kayo? Saturated na ba? Vmax na? Okay pa? Or do you want a break? Another break? Or tuloy na natin? Sige, no reaction. So, let's try to continue muna before we proceed to the next part. So, wait lang. Excuse me. Okay. So, di ba sabi natin, we mentioned that the symptoms related to porphyrias are due to elevations of the intermediates. Tama ba? Diba? That's the reason for the manifestations of your porphyrias. So, as you see here, the degree of overproduction of the intermediates is dependent on the degree of stimulation of the system by endogenous or exogenous stressors that induce your ALAS enzyme. Naintindihan po ba yung statement? Doc, nakaka-overwhelm. Para lang sinasabi natin dyan, when you stimulate ALAS, do you stimulate the production of the intermediates? Guys, when you stimulate ALAS, will you produce the intermediates of the heme synthetic pathway?
Yes po.
Yes po.
Yes, because that's the rate-limiting enzyme. It will keep on making ALA, then ALA to the succeeding intermediates. So when you have an overly active ALAS, the intermediates would also elevate. Okay? Meron niyang clinical correlation. There's a clinical correlation later on, no? That we'll be discussing. So these are the positive regulators of ALAS. First is fasting, glucagon, iron. We discussed this a while ago, right? So your ALAS, no, contains an iron response element, so that presence of iron would stimulate the enzyme. Excessive alcohol and CYP450 inducers. Negative regulators include heme. No, we already answered this. No, heme, the end product of the pathway, would inhibit the synthesis of ALAS. Your glucose and insulin would also inhibit ALAS. Let's try to discuss one by one. Excessive alcohol intake, no, or alcoholism. You're done with the CBL relating to alcoholism, right? Diba? Do you still remember the metabolism of ethanol or alcohol? So, diba, your ethanol is metabolized by different enzyme system. Initially by alcohol dehydrogenase, producing acetaldehyde, then aldehyde or acetaldehyde dehydrogenase, producing acetate. A small portion is metabolized by your catalase, right? Catalase. But in chronic alcoholism or in excessive alcohol intake, no, you will stimulate the liver to produce the enzyme cytochrome P2E1. Cytochrome P2E1 is a member of your cytochrome P450 enzyme system. Naalala niyo yung si cytochrome P450? What is cytochrome P450? Is it a heme protein? Is it a heme protein? Andiyan na naman
Yes
yung mga
po,
ibon.
Doc.
Yes, it is, no, di ba? Cytochrome B450 isa yan sa mga heme proteins together with your hemoglobin, myoglobin, catalase, yung mga pinag-discuss natin kanina. So it's a heme protein. So being a heme protein, does it contain heme? May heme ba siya? Yes. So when you continuously synthesize cytochrome P450, are you consuming a lot of heme?
Yes.
Yes po.
Yes. And if you're consuming a lot of heme, will you deplete the levels of heme? Yes, no? Of course, you will deplete the heme, no? Because you're continuously using it. And when you have low levels of heme, what would happen or what would be the effect into your ALAS? Will it be stimulated or will it be inhibited?
Stimulated.
Low, ha?
Inhibited.
Low.
Inhibited.
It will be inhibited. Kasi ang stimulator, ang stimulator niya, ang inhibitor niya is your heme. Okay? So that low heme can actually stimulate. Ano ba sabi niyo?
Stimulate po.
Okay, so it can stimulate. Tama ba? Do you agree with me? Low heme will stimulate ALAS. Why would it stimulate ALAS? Because you want to make more heme, right? For cytochrome P450. So that excessive alcohol by virtue of decreasing your heme, can stimulate ALAS. So it's a positive regulator of ALAS. The same is true with your cytochrome P450 inducers. Cytochrome P450 inducers are drugs, no? These are the examples, no? Masyadong mahaba, hindi ko na isa-isa yan. I will not go through each one. Basically, cytochrome P450 inducers are drugs that are metabolized by cytochrome P450. So do they stimulate your liver to produce cytochrome P450? Yes. Is cytochrome P450 heme-containing? It's a heme-containing protein. If it continuously use heme, will you deplete the levels of heme? Yes, and that low levels of heme can actually stimulate your ALAS. Okay, so that's the application for excessive alcohol and CYP450 inducers. Next, fasting and glucagon. How does it affect or how does it stimulate ALAS synthesis? Please take note of the figure, no, on your right. When you go on fasting, nagutom ka, your body will try to increase your glucose through the release of glucagon. Glucagon will help you, will give you, no, or will raise your glucose, no, so that you'll have energy. Glucagon will activate its glucagon receptor, which is a G protein coupled receptor that later on activates your cyclic AMP, cyclin adenosine monophosphate. This cyclin adenosine monophosphate activates your protein kinase A. Protein kinase A would stimulate or would produce a transcription factor known as your CREB. When you say transcription factor, no, it facilitates the transcription of a gene. It will tell your gene, okay, please make an mRNA. Okay? So the effect of protein kinase A is the formation of your CREB, no, or your camp response element binding protein that will bind to your camp response element. Binding of two would form another transcription factor. That transcription factor is PGC1 alpha. PGC1 alpha together with another transcription factor, FOXO1, or your foxhead box protein 1, are transcription factors. Together with iron will bind to your iron response element seen in your ALAS gene. So pag nag-bind yung tatlo, when those three bind, PGC1 alpha, FOXO1, iron, when they bind to iron response element, will they lead to the transcription of the ALAS mRNA?
Yes.
Yes, no? You'll have transcription of ALAS mRNA. So in that sense, fasting stimulates ALAS synthesis by virtue of synthesizing CREB that is needed for the synthesis of your transcription factor, PGC1 alpha. I hope nakasunod, no? Kaya ako nilalagyan ng picture, no? Kasi pag binanggit ko lang yan, you'll not be able to follow. On the other hand, no, as you can see here, glucose and insulin are negative regulators of ALAS. Do they stimulate it or do they inhibit it? They inhibit your ALAS synthesis. How will they do that? Glucose intake, whether it's orally or given through IV injection, no, would stimulate the release of insulin, no? Insulin will bind to its receptor, no, your receptor tyrosine kinase, leading to activation, no, of the signal transduction MAP kinase pathway. Have you heard about an enzyme kinase? When an enzyme is a kinase, what does it do? So it phosphorylates, no, proteins, no? And as you can see here, FOXO1, no? When insulin is activated, what happened to FOXO1? It becomes phosphorylated. Phosphorylated form of FOXO1 is the inactive form. Inactive form. If it's inactivated, will there be still FOXO1 that can combine with PGC1 alpha to transcribe your ALAS mRNA? wala na. So FOXO1 will be absent. So in that sense, glucose intake, insulin can inhibit the synthesis of ALAS. Naintindihan po? Konti konti pa. Sige. Don't worry, kung medyo nahihirapan kang intindihin, I'll still be posting a recorded lecture. You can reach me through Telegram, but I think I need to proceed, no? Kasi medyo maubos yung time natin. Okay, so these are the medications that are CYP450 inducers. All right, let's try to apply. Ano ba yung sabi mo, Doc? May application ba yan? Ano ba yung may tutulong pag naging doctor na ako when I become a doctor? Will that help my patient? Will that help me advise my patient on what things to avoid? Yes, no. Let's take this as an example. For example, you have a patient with acute intermittent porphyria. What's the problem in acute intermittent porphyria? Porphobilinogen deaminase. So meron kang block sa pathway. Tama ba? Yes. So so in acute intermittent porphyria, you have elevation of ALA and PBG. Again, what's the manifestation? Neurovisceral cutaneous? Neurovisceral. Okay. So imagine your patient go on a binge drinking or took medications that are CYP450 inducers. They took alcohol, no? Excessive binge drinking of alcohol and CYP450 inducers. What happens to the level of heme?
Decreases po, Doc.
Yeah, it will decrease the level of heme. And that would remove the inhibition of heme to your ALAS synthase. Tama ba?
Yes.
Pag nawala yung inhibition, if the inhibition is removed, will you have overactivity of your ALAS? Yes, no? ALAS is free to do its thing, no? So will you produce more ALA and more PBG?
Yes.
Yes. So you will produce more ALA and PBG. Will that precipitate an attack of porphyria? Will that make the manifestations of your patient more severe?
Yes. Yes.
Yes. So will you advise patients with porphyria to take in alcohol or to take in drugs that are CYP450 inducers? No. Okay. This is also the same reason why we give IV glucose and IV hematine, the synthetic form of heme, in patients with porphyria. Because if you give glucose, if you give heme, synthetic form of heme, will you inhibit ALAS? If you give glucose, if you give heme, yes, because heme is an inhibitor of ALAS. So when ALAS is inhibited, will you still produce the intermediates? No. Will that improve your patient's manifestation? Yes. O, diba? So that's how you apply it. Okay? Any questions up until this point? May tanong po? Sige. Alright. Sige. I think we can have a 2-minute bladder break lang. No? Papahinga ko lang sa gate yung throat ko. Inom lang ko ng water. Bladder break. Okay, 2 minutes. Okay, let's resume. Cameras on na po. Okay pa naman kayo?
Doc, may I clarify
Hmm.
lang po. Hello po, Doc.
Sure, go again.
May I
Go ahead,
clarify
Timothy.
po again po, Doc, as to why we give IV glucose to patients again po, Doc?
Glucose together with your heme, no, are inhibitors of your ALAS synthase. When you inhibit the ALAS, no, you also inhibit the formation of the intermediates.
Okay po, Doc. Thank you.
Okay. All right. So hindi na daw okay, sabi ni Margo. Masakit na sa ulo. Masakit talaga sa ulo ang porphyrins. Trust me guys. Nandyan, umupo din ako na parang ganyan, no? Nakinig din ako. Si Dr. Bravo pa noon ng lecture namin ng porphyrins. So, imagine niyo yun, mas highfalutin words pa. Naka-nosebleed, no? Hirap ka nang intindihin yung topics, no? Sa English, nano-nosebleed ka pa, no? So, yan. Anyway, nag-advanced reading ba kayo tungkol sa topic na to? Kasi the next few sections, no, medyo madugo siya, no? So, if you didn't advance read, no, medyo mahirapan tayo. Pero we'll try to explain it, no, in a simple way. Yun natin kung kaya natin pasimplihan. Anyway, so let's now proceed with the last 4 objectives. Let's discuss the myoglobin and hemoglobin structure and function. First is your myoglobin. So your myoglobin is a single polypeptide chain, no, made up of 8 segments of alpha helices. These designated as A to H, no? So A1, B2, C3, D4, and so on and so forth, no? So your myoglobin is present in the heart and in skeletal muscles, where they act as a repository of oxygen, no, that they later on release, no, during strenuous or active exercise. So dahil sa structure niya, it's not very efficient in delivering oxygen. So ang function niya is to store it, no? So the exterior of the molecule, just like any other proteins, no, are polar amino acids, no, water-loving amino acids. And the interior of the molecule is made up of non-polar amino acids, except for 2 histidine residues, no. One is located at the distal, no, at the E segment, no, otherwise known as the E segment, no, E7, or the distal histidine, that stabilizes the bond between oxygen and ferrous iron. Di ba sa gitna ng porphyrin or ng heme molecule, nandiyan yung iron. So ang function ng distal histidine is to stabilize the bond of histidine with the oxygen, stabilize the bond of oxygen with the iron, no, in your heme. While the other histidine is located at the F segment or the proximal segment that directly binds to iron. Okay? So siya, direct siya yung binding niya sa iron. Ito, yung oxygen, ini-stabilize niya yung binding ng oxygen with the iron in the center of your heme molecule. Comparing it with your hemoglobin. Hemoglobin is a tetramer, 4, no, with 2 different types of globin chain with heme, no. As you can see here, this structure here acting as its prosthetic group. It is a metalloprotein that transports up to 4 molecules of oxygen. And because of its structure, no, it can perform more complex function than your myoglobin. It can transport hydrogen ion. It can transport carbon dioxide, no? And because of its structure, the oxygen binding capacity of hemoglobin is affected by allosteric effectors. Later on, we'll be discussing these, no? Allosteric effectors only applies to oxygen binding to hemoglobin, not with your myoglobin. Okay? So the major form of hemoglobin in adults is known as your hemoglobin A, which is composed of 2 alpha, dalawang alpha, and 2 beta chains. Okay? 2 alpha and 2 beta chains. Okay? Excuse me. Now, let's proceed with globin synthesis. Ito na yung tinahanap kanina. Globin synthesis is carried out by 2 gene clusters. No? One is the alpha globin gene family located in chromosome 16, and the other one is the beta globin gene family located in chromosome 11. Please remember that in normal globin synthesis, you always need 2 globin chains coming from the alpha globin gene family and 2 globin chains coming from the beta globin gene family. Sa normal ha, tatandaan nyo. Okay? Members of the alpha globin gene family includes alpha 1, zeta, and pseudo-alpha. Beta globin gene family includes beta, gamma, delta, and epsilon. In adult hemoglobin, again, what's the composition? You have 2 alpha coming from the alpha globin gene family and 2 beta from the beta globin gene family. Laging ganyan, sa normal. Later on, we'll be discussing the different types of hemoglobin. Makikita natin yan. What I would like you to remember, di ba you'll be having your CBL reporting about thalassemias? Sino yung magre-report ng thalassemia? Meron ba? Okay. So tatandaan ninyo, the alpha globin chain, the alpha chain, has 2 alleles in chromosome 16. Ilang chromosome 16 ba meron tayo? We have a pair, right? A pair of chromosome 16. So in dun sa isang pair, one pair, you have 2 alpha chain. In the other pair, or yung partner niya also has 2 alpha chain, 2 alleles for alpha chain. So how many in total alleles will produce alpha chains?
4.
4 po, Doc.
4. Okay. This is the reason why alpha thalassemia would have 4 classes. 1 gene deletion. Pag na-delete yung isa, 1 gene deletion is known as your alpha silent carrier. 2 gene deletion is your alpha thalassemia trait. 3 gene deletion is your hemoglobin H disease. And 4 gene deletion is your hydrops fetalis. Nasusundan? Yan kasi talaga yung litong-lito ko. Bakit ba 4 yung gene for ano? Why is it that there is 4 genes for alpha chain, no? When clearly, no, we only have 2, no? Because it's a pair, no? So you have 4 alleles for that. Okay? So now let's discuss the different globin chains that we synthesize at various stages of development. Okay? So hemoglobin synthesis begins in the first few weeks of embryonic development within the yolk sac, which is a temporary structure, no? So the major hemoglobin at this point, no, is your hemoglobin goer 1. Hemoglobin goer 1 is made up of your 2 zeta and 2 epsilon chains. O, sinusundan niya ba does it follow our rule that 2 chains comes from the alpha globin gene family? Yes, zeta comes from the alpha globin gene family and epsilon comes from the beta globin gene family. Other hemoglobins that you can produce includes hemoglobin goer 2 and Portland, no? So you can see here, alpha chains, now you can now produce it, and it will complex with your epsilon to form your hemoglobin goer 2. And lastly, your Portland is made up of your zeta and your gamma chains. Now, by 6 to 8 weeks of gestation, no, the expression of these versions of your hemoglobin declines, no? Kita nyo naman, epsilon and zeta declines, no? This coincides with the change of your hemoglobin synthesis from the yolk sac now to your liver. Okay? So sa liver na siya. And at around 10 to 12 weeks of gestation, you now produce your hemoglobin F, your fetal hemoglobin. This fetal hemoglobin is made up of alpha chains, you can see here, as well as gamma chains that can persist, no? That can persist up to 6 months post-natally. Okay? So up to 6 months post-natally. Okay? So, what else do we need to remember? As you see here, right before birth, there is a transition between the formation of beta and gamma chains so that we now begin to synthesize the adult type of hemoglobin, your hemoglobin A, which is made up of alpha and beta chain. And just before birth, as you see here, you can now produce the delta chain from the beta globin gene family. So we can now produce your hemoglobin A2, which is made up of your alpha 2 and delta 2. Okay? So in normal adults, no, 97.5% is adult, 2% is hemoglobin A2, and a few percentage is your fetal hemoglobin. So those are the three types of hemoglobin you can see in adult patients. What else do I need to emphasize? Yun lang. That's it for now. So let's now proceed with the different hemoglobins, the different normal hemoglobins. We've already discussed hemoglobin A, alpha 2, beta 2, 96% in adult, no, 25% in newborns. Hemoglobin A1C, no, is actually a form of hemoglobin A, no, that is glycated, no. So there's a sugar that is attached, no, to your hemoglobin molecule. Specifically, this is a non-enzymatic reaction. Non-enzymatic reaction between the terminal valine residue with the beta-globin chain with your sugar. So silang dalawa, all right? And this hemoglobin A1C can actually be measured, no? And be used as a diagnostic tool in patients with diabetes, no? A value of 6.5%, no? 6.5% would tell you that someone is diabetic. Hemoglobin A2, as I mentioned a while ago, is made up of alpha and delta. Remember, important thing to remember, it is upregulated in beta thalassemia. In beta thalassemia, you have a problem in forming your beta globin chains. No, either a decrease in beta globin chain or complete absence of beta globin chains. So if you don't have beta globin chains, no, what is the beta-like chain that can associate with alpha chain? Delta. Okay. So in beta thalassemia, you will have high levels of your hemoglobin A2, no? Alpha 2, delta 2. Fetal hemoglobin, no, made up of alpha 2 and gamma 2, makes up 75% of hemoglobin in newborns, and they have higher, please remember, they have higher, no, affinity to oxygen than your adult type of hemoglobin. Is that beneficial for the fetus to have higher oxygen affinity than adult type hemoglobin? Yes, no, because the mother, no, what's the hemoglobin that you see in the mother? That's hemoglobin A, no, and your fetus, no, majority of their hemoglobin is fetal hemoglobin. Can they extract oxygen to the mother? I mean, from the mother's hemoglobin? Yes, because they are more efficient, no, in binding your hemoglobin than adult type of hemoglobin. Later on, again, we'll be discussing that. Okay? So ito na yung medyo madugong part, no? We'll be discussing now the different forces, no? What are the forces that holds your hemoglobin together? And what is the effect of oxygen, no, to these forces? First, your alpha and beta chain is stabilized by hydrophobic interactions. This is the dotted line here, no, that you see in this figure. Yan. Okay. And the AB dimers, AB dimer 1 and AB dimer 2, is stabilized by ionic, hydrogen, or polar bonds. No? This is depicted as this red bond in between your dimers. Nasusundan po? Okay. Okay. In the absence of oxygen, we term that hemoglobin deoxyhemoglobin. The two AB dimers, no, has a lot of polar bonds or ionic bonds, making them stiff, no? They would have restricted movement, no? This type of hemoglobin has restricted movement. So we call that as a taut form, T form, or low oxygen affinity form. So in short, the higher the number of ionic bonds, hydrogen bonds, or polar bonds, the more stiff the hemoglobin, the lower its affinity to oxygen, no? So we term that tau or T form. Imagine it like this, no? Yung babae, no? With her arms wrapped around his or her chest. So doesn't want to accept oxygen, no? Low oxygen affinity. So ang tendency dito, the hemoglobin would release the oxygen. Next, in the presence, excuse me, in the presence of oxygen, as you can see here, no? Nilagyan mo ng oxygen, no? What did you notice with the polar bonds or in the ionic bonds? Kumonti ba sila? Did they decrease in number? Yes. Binding of oxygen will dissolve or will remove this polar bond so that the hemoglobin, no, your oxyhemoglobin is more, what do you call this? Is less restricted, is capable of movement, no? And as you see here on the lower portion, it will rotate, no? The AB dimer 1 will rotate at a 15-degree angle relative to your AB dimer 2. And because of this movement, no, this facilitates the formation of the relaxed form of your hemoglobin, also known as the R form or the high oxygen affinity form. Imagine it like this boy with his arm wide open, ready to accept oxygen. Nasusundan po. So parang in short, the more polar bonds, the more ionic bonds you have, the more restricted your hemoglobin, the lower its affinity to oxygen. What's the form that will predominate? The tau form or the T form. The less polar bonds, no, because of oxygen binding, the more movement, no, your hemoglobin can do, the lesser the polar bonds. So the relaxed form or the R form, no, will predominate, or the high oxygen affinity form. Please remember a very important concept. Binding of the first oxygen molecule is the hardest, no? Doon sa 4 na subunit, among the 4 subunit, the first oxygen molecule would have the hardest time to bond or to bind. But binding of the first oxygen will make it easier for the second oxygen to bind, no? Because it's able to disrupt some of the polar bonds already. While the second oxygen, when it binds to the second subunit, it will make it easier for the third oxygen molecule to bind. And the third oxygen molecule would make it the easiest for the fourth oxygen to bind. And we term that as the positive cooperativity effect of your oxygen or your hemoglobin. Nasusundan? Can you follow? Like the konti. Sige. Alright. So paano yun nangyayari? Imagine it like this, no? So this is a heme, no? A heme group, no? Heme group. Ano mo nasabi na heme yan, no? You know, may iron on its center. In the deoxyform of your hemoglobin, deoxyform, with or without oxygen? Without oxygen. In the deoxyform of the hemoglobin, you see the iron placed somewhat lower, no? In comparison to the plane of your heme, no? Yung porphyrin mo dyan, no? Pag walang oxygen, the iron is somewhat lower. This one is your histidine residue, no? Attached. Now, in the presence of oxygen, oxyhemoglobin, oxygen, what did you notice about the iron? The iron now is at the same plane as your porphyrin, as your heme, no? And this would leave now your histidine, disrupting now the ionic pair. So ito yon. This is your heme with iron on its center without oxygen. Without oxygen, the oxyform, what's the state? P state. With binding of oxygen, oxygen would lift the iron at the center, putting it at the same plane as your heme, disrupting now the ionic pair. Pag wala yung ionic pair, anong predominant type of hemoglobin? Relaxed form or taut form?
Relax.
Relax, no? Because you remove some of the ionic pair. Some of the other term for ionic pair is salt bridges. Later on, we will encounter that. So when you remove the salt bridges, this forms your relaxed form or the high or low oxygen affinity form. High oxygen affinity bond. Okay. Medyo ano na, no? Mababa na yung energy. Okay. So next, I hope that's clear. Let's now discuss the oxygen binding to hemoglobin and myoglobin. So you remember, each hemoglobin molecule can bind up to 4 molecules of oxygen. In the lungs, no, hemoglobin is mostly saturated by oxygen. While in the peripheral tissues, no, hemoglobin releases 1/4 of its oxygen. More oxygen will be released, no, in environments with low concentration of oxygen, in low pH, with high concentration of carbon dioxide and high temperature. These conditions are seen in actively metabolizing tissue. In Tagalog, sa lungs, mas saturated ang hemoglobin. Sa peripheral tissue, it is less saturated because you have a lot of carbon dioxide, low pH, low concentration of oxygen. Okay? Don't worry, we'll go through each of those allosteric effector. So now, let's try to discuss the oxygen dissociation curve, no? So the oxygen dissociation curve, napapakagat na ng labi si Micaela. The oxygen dissociation curve, no, plots the percent saturation of your hemoglobin, which is in the y-axis, against the partial pressure of oxygen, which is in the x-axis, and its contribution to the total oxygen component or oxygen content. In discussing the oxygen dissociation curve, we need to remember several terms. First is your P50. P50, parang ito din yung ano eh, yung KM sa ano eh, sa enzyme. P50 represents the partial pressure, please take note, the partial pressure at which hemoglobin or myoglobin is 50% saturated with oxygen. Okay? Next, oxygen saturation. It measures the percentage of hemoglobin binding site occupied by oxygen. So if the hemoglobin molecule is 50% saturated, that's 2 of the subunits occupied, 25, 1, and so on and so forth. Partial pressure naman, on the other hand, is the pressure of oxygen in a mixture of gases, normally 160 millimeter mercury at sea level. Okay. So now that we're able to define several terms in the oxygen dissociation curve, we can now compare the oxygen dissociation curve between your myoglobin and your hemoglobin. Okay, so on your left, no, is your myoglobin dissociation curve. On your right is your hemoglobin dissociation curve. So the myoglobin dissociation curve, as you see here, rises sharply, very steep, no? Eventually leveling off, no? Resembling a hyperbolic curve. Hyperbolic curve. The P50 of your myoglobin is low at around 1 to 2 millimeter mercury, or 2 torr in this example. So it has a low P50 in comparison to your hemoglobin. What does that tell us? Myoglobin is not efficient in transporting oxygen, rather mainly for storing it. Why? Imagine, you already have 1 to 2 millimeter mercury, no, of oxygen, partial pressure. Myoglobin would not release its oxygen content. Nakikita nyo, 1 to 2 millimeter mercury na lang, no? Ang damot niya pa, it would not release it. If you're going to compare it with your hemoglobin, which has a higher P50. So, let's try to discuss myoglobin or your hemoglobin. So, the hemoglobin dissociation curve is different from your myoglobin dissociation curve in that it exhibits several features, no? So the shape of your hemoglobin dissociation curve, is sigmoidal, no, in shape. And this will tell you that hemoglobin dissociation curve exhibits positive cooperativity effect. Diba? Ano ang sabi natin dun sa positive cooperativity effect? Binding of the first hemoglobin would facilitate the binding of the second hemoglobin, the second to the third, and the third to the last oxygen. Okay, so as you can see here, no, at 25% oxygen saturation, pag sinabi natin, when we say 25 oxygen saturation, how many subunits is occupied in your hemoglobin molecule? 1, no? So the saturation or the partial pressure needed for you to saturate 25% of your hemoglobin is around 20 millimeter mercury. That's high. Comparing it with the second, no, subunit, P50. The P50 of your hemoglobin is around 26 millimeter mercury. So from 20, how many partial pressures did you add to saturate the second hemoglobin? Around 6 only, right? So meaning addition of around 6 millimeter mercury would already saturate the second binding site. So does that tell you that the second binding site had an easier binding with oxygen than the first binding site? Yes, no? So that would tell you that the shape of your hemoglobin dissociation curve exhibits a positive cooperativity effect. P50 of your hemoglobin is said to be higher at around 26 millimeter mercury, no? Making hemoglobin efficient in transporting oxygen, okay? Making it efficient in transporting oxygen. Because at around 26 millimeter mercury, it's able to release, no, 50% of its oxygen, okay? So that's the main difference. So now, let's now discuss the different allosteric effectors to your hemoglobin. Allosteric effectors only applies to your hemoglobin and not to your myoglobin, no? Because allosteric effectors affects the binding of oxygen to your hemoglobin, no? The binding and the release of oxygen to your hemoglobin. So these are the factors or the regulators that affects oxygen binding to your hemoglobin. One is PO2, partial pressure of oxygen, partial pressure of carbon dioxide, pH, 2,3 BPG, and temperature. Okay. So on your right, you see the hemoglobin dissociation curve. Paano sabing hemoglobin yan? Sigmoidal in shape. Okay? Please take note. There are two ways on how your hemoglobin, no, can behave. It can either shift to the right or shift to the left, depending on the allosteric effector. Let's first look at the shift to the right. See, a shift to the right means that you have a higher P50. What again is P50? What do we check in P50? The partial pressure in which your hemoglobin is 50% saturated. Tama ba? 50% saturated. So, when you shift the curve to the right, do you also increase the partial pressure? This is the partial pressure. Do you also increase the partial pressure?
Yes, po, Doc.
Yes. So you need a higher partial pressure to saturate 50% of hemoglobin. So what does that tell you? Does that tell you that hemoglobin has higher affinity to oxygen or lower affinity to oxygen?
Lower. Okay.
Lower affinity, because you need a really high partial pressure just to saturate your hemoglobin. Meaning, hemoglobin is snubbing, no, the excess oxygen, no. So shift to the right, no, is mainly seen in the peripheral tissues, no, peripheral tissues, where these conditions are seen, no. You have high PCO2, high hydrogen concentration, high 2,3 BPG, high temperature, and low PO2. Is this beneficial for the peripheral tissues, the actively metabolizing tissues, for the hemoglobin to have low oxygen affinity? Is it beneficial for them?
No.
Oxygen does not want to bind to oxygen. So if oxygen would not bind to oxygen, what would it do instead? It will release oxygen. So is it beneficial for the actively metabolizing tissue that the hemoglobin has low oxygen affinity? Yes, it's beneficial for them. If you have low oxygen affinity, then the hemoglobin would release its oxygen content. Diba? In peripheral tissues, are they actively metabolizing? Yes. So imagine these factors are seen in actively metabolizing tissue. Do they have high PCO2? An actively metabolizing cell. Do they have high PCO2? Yes, it's an end
Yes.
product of cellular metabolism. Do they have high hydrogen concentration? Yes. Do they have high 2,3-BPG? 2,3-BPG is an intermediate of your glycolysis. I know you haven't discussed glycolysis yet, right? Tama ba? So glycolysis is active, no? When cells are actively metabolizing. They are using glucose to form pyruvate, no? So 2,3-BPG, high 2,3-BPG tells us that the cell is actively metabolizing. Low PO2, of course, given if you have low PO2, the lower the oxygen affinity. So the tendency of hemoglobin when it shifts to the left, it shifts to the right, is to release the oxygen. Okay? That's how I remember it. Shifts to the right or release the oxygen. The opposite is true when you shift the curve to the left. When you shift the curve to the left from here to here, what happens to your P50? Will it increase or will it decrease? It will decrease, no? You will have a lower P50. So meaning, you have a lower partial pressure from here to here. You have a lower partial pressure needed to saturate 50% of your hemoglobin. So konting oxygen lang, the hemoglobin will take it. So what does that tell you? Is it a high oxygen affinity form or the low oxygen affinity form? Huh?
Hi po.
High oxygen affinity form. And these are the conditions, no, where you see a shift to the left. Low PCO2, low hydrogen concentration, low 2,3-BPG, low temperature, and high PO2. These conditions are primarily seen in the lungs, no? Tandaan niyo yan. So this is the high oxygen affinity form. Ano yun? P or R? P or R form. A shift to the left. P or R form of the hemoglobin.
R po.
R form. Di ba? You remember a while ago, relaxed form? Wide. Arms wide open. Ready to accept oxygen. High affinity form. A shift to the right, no? Leads to higher P50, low oxygen affinity form. That's that P form or the tau form. Okay, let's try to discuss each of your allosteric effect first. First, pH and carbon dioxide. Sama na din natin ang hydrogen ions. So it says here that hydrogen ions facilitates the reformation of salt bridges. Ano nga sabi natin salt bridges? Is this also the ionic interactions or ionic bonds? Yes, no? This salt bridges protonates the histidine residue, no, in your hemoglobin, facilitating now or converting the hemoglobin to its 10 state. Naalala nyo kanina dun sa AB dimers? AB dimers, what is the bond that held them together? Ionic bonds or hydrogen bonds, otherwise known as your salt bridges. So the more salt bridges you have, what is the state of hemoglobin? Is it in the taut form or in the relaxed form?
Taut.
Taut.
Form.
Taut form. Is that the low or high oxygen affinity form?
Low oxygen.
Low.
Low oxygen affinity form. So if it's the low oxygen affinity form, what will hemoglobin do to the oxygen that it's carrying? Will it release it?
Release.
It will release it, no? And this condition, as I mentioned a while ago, is seen in actively metabolizing tissues, no? That's beneficial for them. Carbon dioxide. Carbon dioxide, by virtue of carbonic anhydrase reaction, I don't know if you've already touched on this, Carbonic anhydrase reaction basically converts carbon dioxide to carbonic acid, eventually hydrogen ions and bicarbonate. So the more carbon dioxide you have, do you have more hydrogen ions? Yes. The more hydrogen ions you have, will you have more salt bridges? Yes. No, because it protonates the
Yes.
histidine residues. And the more salt bridges you have, what type of hemoglobin predominates? The taut or the relaxed form?
Taut.
The taut form or the low oxygen affinity form.
Taut form
Again,
po.
that's beneficial. Yes. Again, that's beneficial for actively metabolizing tissues who's producing a lot of carbon dioxide. Okay. So the last one is formation of carbamates. No, basically carbamates are formed from the reaction of carbon dioxide with the amino group, no, of your hemoglobin forming carbamates. Carbamates are anions, no, anionic compounds. No, this also facilitates the formation of salt bridges. No, so again, protonating your hemoglobin, facilitating the taut form, the tense form, or the low oxygen affinity form, releasing your oxygen. This graph just shows you what's the effect of pH, no? Decreasing pH shifts the curve to the right. Sabi nga natin, when you shift the curve to the right, is that the low or high oxygen affinity form? That's the low oxygen affinity form, leads to the release, no, of oxygen to the tissues. Okay, next, temperature. Temperature, ito lang para madaling maalala, has an inverse relationship, no, with your with the oxygen affinity. So the higher the temperature, the lower the oxygen affinity. The lower the temperature, the higher the oxygen affinity. Okay? May ibang explanation pa yan, medyo mas deep, pero that's how you easily understand it. 2,3-BPG. 2,3-BPG is a product of glycolytic pathway, as I mentioned a while ago. It's an anionic molecule that associate itself, kita nyo dito, associate itself at the center of the molecule. So since it's an anionic molecule, will it form salt bridges, ionic bonds? Yes. So what's the form of hemoglobin that will predominate? Tense or relaxed? The tense state. High or low oxygen affinity form? The low oxygen affinity form. Okay? Ano pa? What's an important takeaway here as well? So your hemoglobin, or sorry, your 2,3-BPG will associate again itself at the center of the hemoglobin, right? So it would form salt bridges with this amino acid. Please take note of the lysine residue here. Okay? Lysine residue is seen in adult type of hemoglobin. But in fetal hemoglobin, instead of a lysine, we see serine. So serine is in your fetal hemoglobin. So between the two, what is the charge of your lysine? Oh, protein chemistry. It's positively charged, right? So will it have a better binding with 2,3-BPG? Yes, because 2,3-BPG is an anion. Lysine is a positive amino acid. But if you change the amino acid from lysine to serine, what is serine? Serine is a neutral amino acid. Okay, so will 2,3-BPG bind more to adult type of hemoglobin or fetal hemoglobin? Adult hemoglobin, diba? Because it has lysine. Now, when you have high 2,3-BPG, will you release oxygen? Yes. Ano ba sinasabi ko? This is the reason why fetus is able to extract oxygen more from the mother. What's the hemoglobin that is coming from the mother? It's the adult type of hemoglobin, right? Your fetus will have the fetal type of hemoglobin. So the adult type of hemoglobin is better in binding 2,3-BPG. When 2,3-BPG binds to your maternal or to adult type of hemoglobin, will your fetal hemoglobin gets the oxygen from your adult type of hemoglobin? Will it better bind oxygen? Guys. Yes, no. Kasi nga, 2,3-BPG cause a release of oxygen. 2,3-BPG binds better to adult hemoglobin than your fetal hemoglobin. Sige, last ano na lang, 2 slides. Sorry, maliit lang ng content dun sa next sa conference. Ano lang naman yun, di ba? Rationalization. Next, carbon monoxide. Carbon monoxide is an odorless, colorless gas, no? That has a very high affinity to hemoglobin. In fact, it is 200 times, no? It binds 200 times more, no, than oxygen, no, into your hemoglobin. So what is the effect of carbon monoxide to your hemoglobin dissociation curve? So ito yung normal, this is the normal. What will it do? It will decrease, no, your hemoglobin dissociation curve because it has a higher affinity to your hemoglobin molecule. But what else can you notice here? It also shifts the curve to the
Left.
to the left. Sabi nga
To the
natin, what is a shift to the left? Higher oxygen affinity or low oxygen affinity?
High oxygen affinity.
High oxygen affinity. Isn't that bizarre? Doc, you're saying that carbon monoxide has higher affinity to carbon monoxide than oxygen. But why is that when you have carbon monoxide, you will have higher affinity to oxygen? That means, no, ang ibig niyang pong sabihin, when you have a hemoglobin molecule, how many subunits do you have? 4 subunits. Let's say you have oxygen bound to 3 of those subunits, and you bind carbon monoxide to 1 of the subunits. That will inhibit the release of this oxygen molecule. Yun yung sinasabi na higher yung affinity niya. Once carbon monoxide binds to your hemoglobin, it will not allow the release of the remaining oxygen. So when you're asked in the exam, what's the effect of carbon monoxide to your hemoglobin dissociation curve? It's downward and leftward. Okay? Right. So last na, promise. Last slide, please. Okay. So parang ganun yung picture niya. Last is your methemoglobinemia. Part kasi ito ng learning objective. Sorry, so hinabol ko. So methemoglobinemia results from oxidation of the ferrous ion present in the heme to ferric ion. So from ferrous to ferric, no? So what are the causes of your methemoglobinemia? So it is due to side effects of medications like sulfonamides, dapsone, antimalarials, antihypertensives like nitrates and anesthetics. It can also be due to an inherited deficiency in your cytochrome B5, no? Cytochrome B5 reductase, otherwise known as your recessive hereditary methemoglobinemia, or due to amino acid substitution as seen here. Normally, take a look at the right side. Normally, methemoglobin can be converted back to its oxyhemoglobin form, the ferrous form, using reducing agents like methylene blue, like ascorbic acid. No? Also, methemoglobin can be reduced back to its ferrous form using the reduced form of cytochrome B5. When cytochrome B5 is used, it becomes oxidized. Now, for you to convert the oxidized back to its reduced form, what do you need? Cytochrome B5 reductase. In recessive hereditary methemoglobinemia, the cytochrome B5 reductase is missing. So can you still convert the oxidized to its reduced form? Hindi na. Will you have reduced cytochrome B5 to reduce your ferric form to its ferrous form? Hindi na din. Okay? So that's for your cytochrome B5 reductase deficiency. So in that case, ano pwede mong gawin? You can use reducing agents, no? Reducing agents like methylene blue and ascorbic acid to convert it back to ferrous form. But ito ang difference niya. The difference between this amino acid substitution is that when you have substitution of amino acids, you cannot reduce, your ferric form to its ferrous form. So even if you have the cytochrome B5 reduced form, even if you have reducing agents like methylene blue and ascorbic acid, you can no longer reduce the ferric form to its ferrous form if you have this amino acid substitution. So manifestations of methemoglobinemia include brownish cyanosis, headache, vertigo, and somnolence because the ferric form is not efficient in carrying and distributing oxygen. Okay? This is different from cyanide, which you also discussed. Tama ba? So cyanide would present with cherry red, no? Cherry red discoloration of the skin. Methemoglobinemia would present with brownish cyanosis. Okay, so I think that's the end of my lecture. Do you have any questions, clarifications?